Today we'll go over the answer key for 331. And our first question is, following an overnight fast, how does your metabolism change? So after waking up, you're not going to have a lot of glucose in your system, so you're going to have a lot of glucagon floating around your system, not a lot of insulin either. So these two hormones, the increase of one and decrease of another, will mean that fatty acids will be released from your adipose tissue. And since you're low glucose, your muscle is not going to want to take up as much glucose because that glucose is going to be used from the brain. Instead, your muscle is going to switch over to fatty acids to try and get energy. And continuing on in question 1, how does it change after a 3-day fast or a 40-day fast? So what happens after a 3-day fast? Your glucagon or your glycogen in your liver is more or less completely gone. It's very low. So you are running low on glucose, and you're going to start upping ketone bodies from your fatty acids. So you're going to increase the amount of ketone bodies, and your brain's going to start adapting to that. In 40 days, your brain is 70% of the energy is coming from the ketone bodies from your fat. You're also going to start reducing the amount of muscle you break down. So you're going to slow that down and start mobilizing your fatty acids to form ketone bodies so your brain can still function. So what's the difference between type 1 and type 2? So type 1 is insulin dependent, so you don't make your pancreatic beta cells, or they have been destroyed. Something's wrong with them. You're not producing insulin anymore, and this is called juvenile onset. While type 2 is non-insulin dependent and maturity onset. So in type 2, you're making insulin, but your body doesn't recognize you're making insulin. Either the receptors aren't working anymore, or it could be anything that just shows, I can't recognize that I have insulin being produced. So that's the difference between type 1 and type 2. What is hyperglycemia and what problems could it lead to? So during diabetes, either through not producing insulin or not realizing you made insulin, you're going to have a higher level of glucose in your blood. This is hyperglycemia. And one of the problems that we talked about that it can lead to is retinal degeneration, glycylation of lens proteins. And just talking about type 1 diabetes, in general, you can have kidney malfunctions, nerve impairment, cardiovascular disease. So you can have a lot of problems due to hyperglycemia. How is cancer metabolism different than normal metabolism? So cancer cells do glycolysis, but they don't really do the citric acid cycle. They're just going to stick to glycolysis even if there's oxygen available. And the reason for this is they want to use the intermediaries to start building proteins, lipids, nucleotides, because a cancer cell is going to want to try and replicate. So they're going to get a lot of glucose flowing towards them. They're going to go through glycolysis, get the energy from that, but use all the intermediaries as well to try and make new compounds for the cell and kind of eschew the oxidative phosphorylation pathway because they want to use everything that they can for building more so than energy. What are the enzymes that add new nucleotides in DNA replication? Those are your polymerases, and they basically work by taking a nucleotide triphosphate, cutting off inorganic phosphate, and sticking it onto the growing DNA strand. In replication, what is a theta structure? A theta structure is the bubble or eye that forms when you're replicating DNA in E. coli. And they form because when you start replicating, you're going to have two different replication forks going from each direction. And you're going to be unwinding this DNA and creating daughter strands. And when you do that, that kind of looks like an eye. So you're replicating DNA in both directions, and you get that theta structure forming from that. Why does the lagging So I should say what, said why. Why does the lagging strand have to be synthesized semi-discontinuously? Why doesn't the leading strand need to do that? All right, so we need a 3' hydroxyl to add DNA to. Our polymerases work by only adding to the 3' end. On the leading strand, we always have a 3' end because of the way the DNA is laid out. But on the lagging strand, we don't always have a 3' end. So we need to start adding these primers to make sure we always have a free 3' end so our polymerase can come and build new nucleotides onto our 3' end. And if you just look at the strands and try to rationalize that in your head, I think it makes a lot of sense. Remember, these DNA strands are reversed, so if one strand is going 5' to 3, the other one is going 5 to 3 in the other direction. What is the purpose of RNA primers in DNA synthesis? Again, we need to add to a 3' hydroxyl. Polymerases cannot just add DNA without a nucleotide already being there. So we need that nucleotide to be there to begin with, and that's going to be RNA. So we always put an RNA primer to start building our new nucleotides onto. The leading strand only needs one primer, while the lagging strand needs multiple primers, and you get fragments of DNA called Okazaki fragments. Alright, and that's all the questions I had for March 31st. Again, let me know if you have any questions. Otherwise, I'll see you in the next video.